Thank you very much. So welcome everyone to this lecture, which is on biological mass spectrometry. So my name is Holger Kramer. I'm the head of mass spectrometry at the MRC Laboratory of Molecular Biology. I've started here just under 6 months ago. So a lot of the data that you will see is from my previous positions, but there is actually some data from the LMB as well. I would suggest that if you've got any questions, please feel free to put them into the Q&A, and I will come to these at the end of the lecture. So just starting off with an overview of an outline of the lecture. So I'll start on a brief overview of types and performance of different mass analyzers. I'll then introduce a few concepts in mass spectrometry, and then also show the equipment we currently have in the MRC LMB Mass Spec facility. And from there, I'll go into different ionization methods, mainly really to highlight two different methods, which are electrospray ionization and the MALDI Imaging method, because these are the two methods that really were instrumental to being able to analyze complex biological molecules like proteins. And then I'll go into a few application areas of these methods. So this is a really simplified scheme. And what that shows is that any mass spectrometer minimally needs to have 3 elements to it. And these are an ion source for the formation of ions, a mass analyzer where the ions are separated, and a detector for detection of ions. And here's a few examples of popular approaches to the ion formation or ionization. So the most important ones, as I just mentioned, are electrospray ionization and matrix-assisted laser desorption ionization for proteomics and for biomolecules generally. But it's quite helpful to contrast these two methods that were there before to really see what these newer methods added to the field when they were introduced. Then there'll be a few words on various types of mass analyzers. For what we do in the lab, the quadrupole, time-of-flight, and Orbitrap analyzers are most important, but we also work with ion trap mass analyzers. And finally, I'm not actually going to go into sort of the specifics and merits of ion detectors. That's probably going to be beyond what was likely or what was reasonable to cover in an introductory lecture. And as I mentioned before, these two soft ionization methods, ESI and MALDI, they were really what allowed mass spectrometry to be applied to biomolecules from nucleic acids to proteins and other polymers. So I wanted to start with just showing an example of a mass spectrum. So if you've not come across this before, then it's just good to sort of establish a few of the basics. So generally in a mass spectrum, on the x-axis, we're looking at mass over charge ratio. So we're not measuring mass directly. It's actually the mass relative to the charge of the ion that is detected. And at the y-axis, we've got an intensity, and that could either be an absolute intensity as it's shown here, or relative to intensity where the whole spectrum scales to 100%, where 100% is the most intense peak in the spectrum. Just a few of the kind of terms and terminology that I'm going to use, and I'll come back to these, so don't worry about sort of trying to keep up with these straight away. But basically, the mass range limit essentially is something that's specific to the mass analyzer, and essentially specifies the M over Z range, the range of M over Z values, which can be measured within the mass analyzer. Then another important concept is the analysis or scan speed. So essentially what that means is the speed at which a specified mass range can be covered by the mass spectrometer, and they can be expressed in mass units per millisecond. So essentially, the higher that scan speed, the better the mass spectrometer will be able to keep up with maybe very complex samples, or if there's a changing picture thanks to chromatographic separation. Transmission will not go a lot into that, but it's worthwhile to keep in mind that not all of the ions that are being generated in the ionization source will necessarily enter the mass analyzer and find their way all the way to the detector. So that will have a direct impact on sensitivity of the method. And then two concepts that are somewhat related. One is the concept of mass accuracy. So essentially the question of how accurate is the mass that is measured in the mass spectrometer compared to the theoretical or expected value for a particular type of molecule. And that's typically not expressed as a percentage. Essentially, it's a fraction of, what's the fraction of that deviation or delta M mass value between the theoretical and the measured mass. But it's normally expressed as parts per million. And that's just an expression of the fact that most mass spectrometers are relatively high in their accuracy, and therefore we need a very sensitive scale. So the part per million scale provides that. And then finally, the resolution. Now, resolution, there's different definitions for mass resolution, but the one that we will be using here is essentially where the mass resolution is defined as the width of the peak or delta M value at half height. So at 50% of the intensity of the mass peak. And what you find is with increasing mass resolution, the width of that peak decreases. And we'll look at that effect in a minute as well. So, so just to get a bit of a feeling for how these types of performance characteristics change with different types of mass analyzers. So I've just provided a list of 4 selected mass analyzers here that are relevant to the work that we do in the facility. And they're the quadrupole, the ion trap, the time-of-flight device, and a specific version of that, which we'll go into, which is a time-of-flight device with a reflectron, and then the Orbitrap that we use for a lot of our bottom-up proteomics work. And essentially, the take-home message here is that there is a variety of different characteristics, and the most important differences are in terms of the mass resolution. So the quadrupole and ion trap would normally be viewed as low mass resolution mass analyzers, whereas time-of-flight and Orbitrap are high mass resolution mass analyzers. And it's not a coincidence that the low resolution instruments will generally have a lower mass accuracy they can achieve, whereas the higher mass resolution instruments will be able to achieve a higher mass resolution as well. Just to show an example of the effect of the mass resolution on the MS data, what is shown here is a signal that could come from a particular peptide, let's say a triptych peptide of a digested protein sample. And in a low mass resolution instrument, such as the ion trap, you just start to see some kind of separation of isotopic peaks, where we're able to see that the peptide ions analyzed in the mass spectrometer contain different numbers of heavy isotopes. Whereas at a much higher resolution, such as that achieved in an Orbitrap mass analyzer, you can see very clear separation of each of the isotopic peaks. And the first of this distribution of peaks, that's referred to as the monoisotopic peak. And then we've got the C13 peak, or the +1 peak with one heavy carbon 13, or generally one heavy isotope, and so on. There's the next one with 2, 3, and 4 heavy isotopes respectively. Essentially, the bottom line here is, in order to be able to resolve this kind of isotopic peak pattern, we require sufficient mass resolution. So just to sort of make this a bit more real and to show what we're working with in the facility, here's an example of 3 types of Orbitrap containing mass spectrometers. These are the Q-Exactive series instruments, and they are actually a type of hybrid instrument. So they contain 2 analyzers, a quadrupole and an Orbitrap. And the combination of these 2 types of mass analyzers allows you to perform more complex type of experiments. So for example, the quadrupole could be used essentially as a mass filter to select a particular ion, and then perform an MS-MS or MS fragmentation experiment. Here's an example of 2 mass spectrometers that also contain Orbitraps, but in addition to the Orbitrap mass analyzer, they contain an ion trap as well. And again, that gives you more flexibility in terms of the kind of instrument, the type of experiments that can be performed with this type of instrument. And it actually means you can optimize the instrument time, where different parts of the mass spectrometer can operate in parallel, doing different things as part of the instrument duty cycle. And the application areas for these types of instruments would be primarily protein identification, quantitative proteomics, and there will be another lecture next week on quantitative proteomics specifically, as well as the identification of post-translational modifications, or PTMs, and fossil proteomics is a good example of that type of application. And then also structural proteomics experiments. So the kind of structural proteomics experiments we would perform on these instruments at the moment typically would be cross-linking mass spectrometry. experiments. I will not go into this in detail in this lecture, but again, in the following lecture, this will be mentioned. And then going into the time-of-flight instruments. So here are two types of quadrupole time-of-flight instruments, where again, a quadrupole has been combined with a time-of-flight mass analyzer. This one here, the micromass LCT, as well as the Xevo G2, they are both used for intact protein mass measurements. Slow on the follow-up here. So basically, on the left-hand side, we've got three time-of-flight instruments. The two in the middle combine a quadrupole with the time-of-flight mass analyzer, and the LCT only has the time-of-flight mass analyzer. And then the application areas would be intact protein mass measurements, and we do a fair bit of this type of work. So that would be typically performed after the protein has been purified in order to characterize it fully. And then the SYNAP we typically use for a different application that's referred to as hydrogen-deuterium exchange mass spectrometry, or HDX. And that is another structural proteomics approach, which allows you to look at accessible residues on a protein surface that are able to exchange with the solvent. Now, I will go into the MALDI, or matrix-assisted laser desorption ionization process a little bit later. I think at this point it's sufficient to say that this is a very versatile technique, so it can be used for protein analysis with peptide mass fingerprinting. It can perform MS fragmentation, but we've also used it for lipid analysis, oligonucleotide analysis, and a whole host of other things. Overall, it's fair to say that the MALDI process is not the sort of the mainstream of proteomics analysis these days. So all of the other instruments that I've shown on the previous slide and this slide, other than the MALDI instrument, they all use electrospray ionization. And the main reason for the success of electrospray ionization really has been the fact that it can be coupled directly to liquid chromatography separation. And that makes it a very powerful approach. Now, what I will do now is I will cover three ionization approaches. And essentially, the first one is mainly to highlight what the novelty was of electrospray ionization and MALDI ionization when these were introduced. And essentially, these two techniques really enabled then the application of mass spectrometry to the analysis of biological molecules. So what is shown here is a diagram showing an electron ionization source. Sometimes in the older literature, this is also referred to as electron impact ionization. And you may have come across this at A levels. So in A levels, whenever mass spectrometry is mentioned, quite typically it's always electron ionization, and that's probably largely for historical reasons. It's a method that has been around for a long time. And therefore still features in the school curriculum very heavily. It's still relevant today, mainly in the context of GC-MS instruments, so combining gas chromatography separation with mass spectrometry analysis. So looking at the diagram of this ionization source, it basically combines a heated filament that is used as an electron emitter. So essentially, by having the current flowing through the filament and then applying another perpendicular potential to that, the heated filament will eject electrons, and these electrons are accelerated towards an anode electrode for electron discharge, and that essentially generates this electron beam here. And that electron beam will then collide with molecules in the gas phase. So for these techniques, the molecules to be analyzed need to be in the gas phase already. And collisions of electrons and molecules in the gas phase will then lead to ionization. And those ions generated in the source, they then have to be extracted, focused, and accelerated to the analyzer. And in GC-MS instruments, this typically would be a single quadrupole for analysis, so a low resolution mass analyzer. As I mentioned, this is often coupled with GC. That actually makes GC-MS a very affordable technique. So a GC-MS instrument that might set you back about £50,000 or thereabouts. The other instruments we just looked at, they tend to be significantly more expensive than that, often 10 times or more expensive than that kind of ballpark figure. One of the potential drawbacks of electron ionization is it leads to extensive fragmentation in addition to ionization of molecules, and we'll be looking at a spectrum on the next slide for that. Often you still observe a molecular ion peak that results from essentially shooting away a single electron from the molecule, resulting in a radical cut ion. As I mentioned, it's often observed, it's not always observed. So if the molecule is relatively sensitive, that might lead to extensive fragmentation and no formation of a molecular ion peak. And while the extent of fragmentation is dependent on the energy level of the electrons, the ionization efficiency is also affected. So one could think that, well, can't we just use electrons of a certain energy to essentially make this whole process a bit softer in terms of ionization? But if we reduce the energy of the electrons, the whole efficiency of the ionization will also be affected and therefore will be decreased. So in terms of the wavelength of the, so it's worthwhile remembering that, of course, electrons can be viewed as a particle or as a wave. And in terms of the wavelength of these electrons, we've got this relationship of the wavelength lambda being the ratio of the Planck constant h to m times v, where v is related to the energy of the electrons. Essentially what I wanted to point out here is that in most ESI-MS experiments, electrons of an energy of 70 electron volts are used. And that's not a coincidence. Essentially, that wavelength corresponds to a bond length that is similar to the type of bonds that need to be excited for either ejecting an electron from that bond or breaking bonds in that type of molecule. So the typical carbon-carbon bond length in organic molecules would be about 1.5 angstrom. Just to show an example of the result of that kind of ionization process. So if we ionize phenylalanine in this way, then a spectrum that looks like this would result. And this spectrum is retrieved from a spectral library. And we see that at the very low intensity, but of the highest mass unit here, we see the intact molecular ion, 165, but then we see a whole series of other ions as well. And these represent fragments where the molecule has lost various groups. So here, for example, the carboxylic acid group has been lost. Here we see the R group of the amino acid that distinguishes different amino acids from each other. So here a benzyl group from phenylalanine, or here essentially what is left of the molecule after the benzyl group, after the R group has been lost. So we see ionization and extensive fragmentation of the amino acids in this approach here. Now, one advantage of electron ionization is that the fragmentation pattern observed is relatively independent of the exact source design. So that is something that's somewhat different to some of the sources used for the milder ionization techniques, such as Electrospray or MALDI. Here, the exact source design is not too critical. The shape or the type of pattern of fragmentation we observe here is mainly dependent on the nature of the molecule itself. And that is quite helpful for the generation of spectral libraries, where you can experimentally determine the spectra of a whole range of different molecules of interest, and then they can be used as a library to try to match experimental spectra of unknown molecules too. So now moving on to electrospray ionization, or ESI. So that is a technique that was introduced by John Fenn, and he shared the Nobel Prize in Chemistry in 2002 for that technique. So essentially what that entails is having a needle that has essentially a channel in the center, and through that channel we can flow liquid. And then the needle either has to be conductive, so it could be a metal needle, or it has to have a conductive coating so that we can apply a high voltage to it. And when we apply that high voltage, what we observe is this phenomenon here, that there's a jet of liquid formed, and this jet of liquid ultimately forms to a plume of droplets of the liquid that's flowing into the needle. Generally, electrospray ionization is a much softer ionization technique. We'll look at the mechanism of how it actually works on the next slide. And because it's a soft ionization technique, it allows to keep very sensitive biomolecules such as proteins intact. There's really two things happening in this process here. molecules are ionized and they're transferred from the liquid phase to the gas phase at the same time. So it's essentially moving something that's in the liquid to the gas phase. And being in the gas phase is really a prerequisite for any molecule to be analyzed in a mass spectrometer. And that's a critical step because, you know, proteins don't normally evaporate, they're not volatile. So therefore, finding a way to move them into the gas phase without destroying them by heating them up, for example, is quite an important point here. And then it's relevant mentioning that the polarity can be either positive or negative polarity, depending on what voltage we apply to this dip. Correspondingly, with positive polarity, we produce cations, and with negative polarity, we will produce anions in the process. Another important characteristic is that it's quite common to form multiply charged ions. So the cations or anions are not necessarily singly charged. They could contain a number of charges, and what that number is, that will depend on the overall mass of the molecule and the functional groups present in the molecule that's being analyzed. And one of the real strengths of the electrospray ionization approach really is the fact that it can therefore be coupled directly with methods that allow you to separate molecules in the liquid phase. So high performance liquid chromatography couples extremely well and therefore makes the combined HPLC electrospray ionization approach very powerful for the analysis of complex mixtures. And that's really what enabled the inception of mass spectrometry-based proteomics. So to look at this a little bit more mechanistically, so here we have a diagram of the process, and the application of the high voltage really is what leads to the formation of this Taylor cone. It's essentially, as we apply charges to the liquid, equal charges will repel each other, and that leads to essentially initially this Taylor cone forming, and then when a critical voltage is reached, it leads to the start of the spray formation. And initially these can be quite large droplets on the size of micrometers. And then we're able to reduce the size of these droplets through evaporation of solvent from this, so to transition from these micrometer to nanometer sized droplets. And then the mechanism for then how this transition from liquid phase to gas phase actually is performed, that has been studied both experimentally and through simulations. And essentially there's different models that have been developed for how this can be explained. So the first model here is this ion evaporation model shown in A. And there the idea is that as the size of the droplets reduces when solvent molecules evaporate, the charge density on the droplets increases. And if the charge density reaches a critical value, then at least for small molecules, it's possible that these analyte molecules might detach from the surface of the droplet. Essentially, once the force that's due to the repulsion between the equal charges overcomes the surface tension of the droplet, we can pick off an analyte ion from the surface of this droplet. So in small molecules, that's very much relevant. However, if you look at large molecules, such as an intact globular protein, it is probably less likely that will actually be able to perform successfully. And in this scenario, a different model is likely to be predominant. And this has been referred to as a charge residue model. So here the idea is that the size of the droplet decreases, and on average, most droplets are likely to contain a single molecule, a single protein molecule, and that eventually all of the solvent molecules are stripped off, and the charges are left on the surface of the protein. And then finally, there's a third model shown here, and that's been termed the chain ejection model. And that's more relevant for either disordered or denatured proteins. And whether proteins are denatured or not also depends on the conditions in the droplet, so in the liquid phase. And in this model, the idea is that this disordered denatured protein that may again be present not in the center of the droplet, but accumulate on the surface of this droplet. And then eventually, when charge density is high enough, ejection of that chain from this droplet can occur. And just to show what that looks like in practice when this process is applied in the mass spectrometer, that's just shown here. So in addition to the electrospray needle and the high voltage, there's a few other things that are required. One of these is a desolvation gas. So this would typically be nitrogen heated to a certain temperature. And that nitrogen flow essentially is opposing the flow of the ions towards the ion guide due to the voltage applied. And that flow of nitrogen aids with the evaporation of solvent molecules from these droplets. And then the other thing that provides a force for these droplets to go into the mass spectrometer, and ultimately the ions to go into the mass spectrometer, is the difference in pressure. So on the outside, there's atmospheric pressure, and then there's various vacuum stages into the mass spectrometer that may reach from sort of a few millibars at the early stages down to 10 to the minus 7, 10 to the minus 10 millibars in the inside of the mass spectrometer where the mass analyzer is based. And again, we've talked about the different models for the ion desolvation, but it's important to mention that it's possible to apply conditions which really then strip off all of the solvent molecules from the surface of the protein and then have a charged protein ions in the gas phase. So what is shown here is an example of this from a measurement we did at the LMB of an intact protein that was analyzed by LC-MS, so liquid chromatography coupled to mass spectrometry. And what I'm pointing out here is that what we see is this whole charge state distribution. So each of these lines, each of these peaks corresponds to a particular charge state. And to not make the whole spectrum too crowded, I didn't try to label all of these, but essentially what we see is a consecutive series of charge states. So this ion here that would have a charge would have 11 positive charges. So from here you could count 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, and so on. And because what we're analyzing in the mass spectrum is the mass over charge ratio, as we increase the charge, the mass over charge ratio will decrease so that the high charge ions, they will be at this end of this distribution, and the lower charge ions at the higher M over Z end. And it's worthwhile also observing that it appears that the protein has some kind of preference for there's certain charge states that are more abundant than others. And what this distribution looks like generally depends on both the functional groups, amino acids that are present on the protein in the protein sequence, but also the overall mass, where the general rule of thumb would be the bigger the protein gets, there might be a different distribution of charge states. However, the chemistry of the amino acid residues is important. So you can have two proteins of the same mass and having different charge state distributions depending on the basicity, so the pi value of the protein, and leading to different distributions. So what I'm showing here is now if you apply mathematical operation, and it's in this case this maximum entropy method or max end algorithm, you're able to convert this whole series of different charge states into the singly charged ion. And that essentially will significantly facilitate and simplify the analysis. If the instrument is well calibrated, then you would expect to see a very close match between the expected mass and the observed mass. And in this particular experiment, that was indeed the case. So here, the expected mass is essentially calculated just from the primary sequence of this protein, and the observed mass was measured in the experiment. Now, just one word or one slide on the coupling of liquid chromatography to electrospray ionization. So this essentially is a critical step to enable mass spectrometry-based proteomics. And what's shown here is a scheme essentially of an HPLC system, shown on the left-hand side, where we use a switching valve to essentially coordinate two different types of liquid chromatography columns. These two columns, they help at different flow rates. So pumps A and B, they produce a flow that flows through the analytical column. And this analytical column has a very narrow diameter of 75 micrometers. And on the other hand, this pre-column or trapping column will have a wider diameter, will be operated at a higher flow rate. And the reason why this is being operated in this way is that the trapping column essentially can bind any of the peptides present in the sample, and they can be loaded at a higher flow rate of 10 to 20 microliters per minute. So it means we can inject a sample of an average, 5 to 10 microliters within a reasonable timescale. Essentially, that trapping leads to desalting and another purification step of the peptides. Then after this switching the valve, it's possible to then direct the flow from pumps A and B through the trapping column, eluting peptides with a gradient of increasing organic solvent content, typically acetonitrile. And then as these peptides are eluted, they then are separated by the hydrophobicity on the analytical column. And then at the end of that process, we have the electrospray ionization interface that we just discussed. Now, what's the overall purpose of sort of scaling down this process for these very low flow rates, so that the flow rates for pumps A and B is in the range of nanoliters per minute? 250 nanoliters per minute would be fairly typical. And the reason for doing that is essentially to be able to work with very limited amounts of sample, as might be obtained from experiments like protein interaction experiments to look at protein interaction partners by immunoprecipitation and then mass spectrometry analysis. And when these sample amounts are very limited, scaling down the whole process to low flow rates and very thin column diameters allows us to analyze these very limited sample amounts. I just wanted to mention the MALDI process, or matrix-assisted laser desorption ionization. That's shown in this scheme over here on the left-hand side. So essentially what happens here is we've got a sample which is present in a so-called matrix, and we'll go into what these matrix molecules might be. And we leave this to dry down and crystallize on a surface, typically on a metal surface on the sample holder. And that leads to crystallization of the matrix. So typically the matrices that are used in MALDI will crystallize very well. And then what we do is we use a laser beam, could be a nitrogen or solid state laser in the UV range. And when we shoot with the laser onto this sample spot, that leads to two things, again, ejection of molecules into the gas phase and ionization. And once this ionization has been performed, we can accelerate the ions with various electrical fields. And I'm not going to go in massive detail through this sort of schematic diagram on the right-hand side here. It's suffice to say that essentially there's probably a mixture of mechanisms in operation where either preformed ions in the matrix are ionized through this rapid heating that is performed when the laser hits the matrix, and the matrix molecules absorb the energy of the laser. But also alternatively, some ionization will occur in clusters in the gas phase immediately after hitting this spot with the laser energy. And again, the same Nobel Prize in Chemistry 2002 was awarded for the MALDI process as well, to Koichi Tanaka, so Japanese scientists, and Paris and Hillenkamp. They actually developed the method in its current form, but yeah, it's worthwhile remembering Nobel Prizes are awarded usually for the first inception of the concept rather than the final refinement of the methodology to its final stage. Now, just showing some examples of the matrices that are typically used in MALDI. So there's various abbreviations. What all of these have in common is that, well, what the majority of MALDI matrices have in common is that they tend to be acidic molecules with carboxylic acid groups, such as CHCA, DHB, or this hydroxypicolinic acid. Now, some of these look a little bit exotic, and you may wonder how did anyone come up with using these for MALDI ionization? So that has been largely quite an empirical process of optimization. So a large number of molecules were tested for their ability to promote MALDI ionization. What they have all in common, though, is they all tend to have extended pi electron systems. So they all tend to be aromatic compounds with maybe other additional functional groups that extend that pi system. Yeah, all of the matrices, or the vast majority of the matrices used in positive ionization mode tend to have acid groups as well, again, assisting in formation of positive ions, cations in the MALDI ionization. And here on the right-hand side, this alternative DAN, or 1,5-diaminonaphthalene matrix, that's often used in negative ionization modes for various types of experiments. And here's an example of this type of target plate. So you can spot up to 384 different samples on this plate. And then thankfully it's possible to do automated acquisition, or alternatively, if there's a limited number of samples deposited on the plate, manual acquisition of spectra from this plate. So just to show how then that kind of mass spectrometry can operate, I just wanted to show a diagrammatic scheme of a MALDI-TOF instrument. So essentially, we've got this sample holder on the left-hand side within the ion sort of the mass spectrometer. And what we then do is we shoot with the laser onto the sample holder where the matrix mixed with the analyte has been deposited. And then when we do that, that leads to this formation of an ion plume. This ion plume, from this ion plume, we can now accelerate ions with a high voltage of about 20,000 volts. And they are then accelerated to this grid shown here. And then after that grid, and essentially that voltage applied to the grid is the acceleration voltage, or Vs. And essentially we can control the energy put into the ions at this stage by controlling the electrical potential applied here. And then after the grid, after the acceleration of the ions, we've got this drift path here, which is also referred to as a field-free region. So the idea being that we've put in energy into the ions and converting potential electric energy into kinetic energy. And the kinetic energy for all of these ions should be constant, and is given by the equation that you would have come across previously, 1/2 mv square. And the central condition is all of the ions accelerated will have the same kinetic energy. However, that now implies that ions of a lower mass, they should be faster, and ions of a greater mass, they should be slower as they then fly down this time-of-flight tube. And then ultimately, when we take the different equations together, what we can work out is if we have determined the time of flight, the only thing that's unknown to us in this experiment is the mass of a charge, or m/z value, that can be worked out in the experiment. So this version of a time-of-flight mass spectrometer is known as a linear time-of-flight mass spectrometer. There is another version that is also realized in the instrument that we have in the facility. That is this Reflectron Time-of-Flight Mass Spectrometer. And I just wanted to show what essentially is different in this process now compared to what we've done previously. So because we've got this electrostatic reflectron at the end of the flight tube, we're able to reflect ions and essentially make them hit a different detector that's actually on the same side as the sample holder is on the other end of the flight tube. And what that helps us to achieve is the following. Even though I stated earlier that the kinetic energy of all of the ions should be the same after the acceleration, in reality, there is still some dispersion of kinetic energies. And this dispersion will lead to broadening of peaks in the mass spectrum. However, by installing this reflectron at the other end of the flight tube, we achieve two things. Firstly, the actual flight path of the ions is now longer than would have been in the linear experiment. But that's just a minor effect. Adding some length to the time of flight tube is useful. But what is more important here is that ions that have slightly higher kinetic energy, they will penetrate slightly deeper into the reflectron, whereas those with lower kinetic energy, they will penetrate to a lower depth of the reflectron. And ultimately, the two flight paths have a slightly different length. And what that leads to in the end is that the two ions, they should then hit the reflectron detector at the same time. So essentially, we've found a way to correct the distribution of kinetic energies, the dispersion of kinetic energies, and by doing that, we're able to sharpen up the peaks and the mass resolution achievable in the device. Now, whenever you do something like that, there's normally some kind of trade-off, and that's shown here. But in the linear mode, you have maximum sensitivity, but a somewhat lower mass resolution. Whereas with a reflectron, we've increased the resolution achievable, but we will lose some sensitivity by performing this process. Now, what I now want to show is an example of how this can be applied. And actually, what I wanted to show here is a fairly unusual application. So I'll just go through this step by step. So so MALDI can be applied to a technique that's called mass spectrometry imaging. And the idea here is that we want to be able to look at the distribution of molecules across the surface of a tissue. In order to do this, we've taken thin sections of tissue and then sprayed a MALDI matrix onto this. And I'll show the exact workflow applied here on the next slide. But what we're now able to do is we still will use a laser, a UV laser, to shoot on the sample stage. But after performing the ionization, we're then able to couple the ions generated to a different type of mass spectrometer, in this case, an Orbitrap-based mass spectrometer. And the reason why we're able to do this is because now we're using a different version of the MALDI process. So on the previous slide, what we looked at is vacuum MALDI, where the sample is in a high vacuum. Whereas here, we've got a situation where the sample is kept at atmospheric pressure. And that version of the process is also called AP-MALDI. And that allows us now to not just couple the MALDI ionization to almost any type of mass spectrometer, but it also allows us to now analyze a sample that is kept at atmospheric pressure and basically analyze a sample that would not cope well with vacuum conditions. And now what we do is we essentially scan all of the tissue, and that this can be a slow process. So recording an image of this type that could take several hours. And at each spot where we shoot with the MALDI laser, we obtain a mass spectrum. And now what we can do is we can show the distribution across of different ions across the tissue in different colors. So obviously, this is not a microscopy technique. We're not actually working with light here. We're just using different colors to show the distribution of different mass ions across this tissue. And it's worthwhile mentioning that typically when performing this, the molecules that are either most abundant or which ionize very well in MALDI ionization tend to be observed most predominantly. And typically a lipid species fulfill both of these criteria. So lipids tend to be reasonably abundant across tissues and tend to ionize very well in MALDI ionization. And here there's sort of three different molecules with different spatial distributions shown on a thin section of the mouse liver. And then here we've got a region of the mouse liver where we've got one of the ions seen at a higher concentration compared to the rest of the tissue. So just to show how the sample preparation then is performed, and it's worthwhile mentioning this is quite a specialized process. So when performing mass spectrometry imaging experiments, typically you would have a specialist focusing fully on mass spectrometry imaging to perform these type of experiments, and that it includes both sample preparation, typically with cryosectioning to get a thin section of about 10 to 20 micrometers. And then in addition to performing the MALDI experiment, you would also want to have an optical image. So you could use a hematoxylin eosin stain in order to get some kind of contrast from this tissue. And the example here was essentially mouse liver, where on the left-hand side, this was a model of fatty liver disease, and on the right-hand side, a healthy wild-type control mouse. And then the sections that we observed that we stained here, that is essentially an adjacent section to the one we will use for the MALDI imaging experiment. We then coat the thin section with a MALDI matrix, and a MALDI sprayer is normally used, where you have a solution of the MALDI matrix being sprayed onto the tissue. And then that whole prepared tissue is set up on an acquisition stage. And then we perform the MALDI experiment, where the laser has to scan across the tissue, or in this case, across both tissue sections here. So that's usually the slow step in the whole process, acquiring data across the whole tissue. And then in the end, it's important to mention that data processing is required to then essentially generate these grayscale images, where you show the distribution of a single ion across the whole tissue. Now you could generate an image like this for top 100 or top 200 most abundant ions observed across the tissue. And then you could pick certain ions that maybe have a differential distribution. So here there's 3 ions shown in red, green, and blue. where the red ion was observed sort of very much at the edge of the fatty liver here. So it's not exactly clear to us what that is. We think it could be some kind of oxidation process happening at the edge of the tissue, or some kind of other conversion of a metabolite or lipid species. Then we've got an ion that was very abundant in healthy tissue, but of much lower intensity in the diseased tissue. And then in blue, another ion that tended to be more abundant on the diseased fatty liver, but not very abundant on the wild type liver. Yeah, so now moving on to the application of mass spectrometry to tandem mass spectrometry experiments. So essentially protein analysis and protein identification in proteomics experiments. And just showing you that MALDI-TOF is not just used, or MALDI is not just used for this slightly unusual MALDI imaging application, but here maybe a more standard application, where what we did here was investigate the conversion of a substrate peptide by an enzyme. This enzyme here is called ASP-H, which is a hydroxylase. And this type of hydroxylase will use oxygen in order to convert asparagine or aspartate residues in the polypeptide sequence to hydroxyaspartate or hydroxyasparagine residues. And what we were interested in observing this, and in this particular project, what we found was that when the substrate had a particular disulfide bridging pattern here, shown where these two cysteine residues here formed the disulfide bridge, as well as these two cysteine residues here formed the disulfide bridge. We did not observe conversion of the substrate peptide to product. The low intensity signal here, that was an impurity present both in the original starting material and after an attempt at conversion. Whereas if we had the correct disulfide bridging pattern, as shown on the right-hand side, we did observe hydroxylation. So after a certain incubation period, we have largely converted the starting peptides to the hydroxy aspartate version that was not observed in the starting molecule. So this is an example of application of MALDI-TOF to observe conversion of a peptide substrate. Now, what I wanted to show you now is the ability of mass spectrometers to not just look at intact peptide masses, as shown on the previous slide, but also to actually sequence peptides and obtain fragmentation spectra. And the example shown here is MS fragmentation, or collisional induced dissociation, CID, as can be performed, for example, in a triple quadrupole mass spectrometer. And what happens here is, after electrospray ionization, we've got ions in the gas phase, which are now termed precursor ions. We can accelerate these into a region of the mass spectrometer where we have a certain concentration of nitrogen molecules, and that leads to collisions with the nitrogen molecules, and each collision will transfer kinetic energy. That will happen several times until a certain threshold is reached. And when there's enough vibrational energy in the precursor ion, we observe fragmentation to various product ions. And essentially what this is an internal heating mechanism where each of the collisions with gas molecules will be translated into vibrational energies until bonds break. Now, this is shown here. So there's a whole nomenclature around the fragments of polypeptides that can be generated in this manner. If we've got an N-terminal fragment that results from breaking at the actual amide bond or peptide bond between the carbonyl and the nitrogen, then we generate these B ions, such as the one shown on the left-hand side. And they can be unstable, so B ions can degrade further to form A ions, shown below here, essentially through a loss of carbon monoxide. However, if we break this bond, we would expect that not only is there an N-terminal fragment that is formed, but there's also a C-terminal fragment formed, and that C-terminal fragment, that is referred to as a Y ion. If we've got, rather than a whole polypeptide chain still connected, if we only have a single amino acid, that is also referred to as an ammonium ion. And that can be indicative of the presence of that particular amino acid in the sequence. Now, in most cases, the likelihood of these fragmentations occurring at any given peptide bond will not necessarily be exactly equal. It will depend on the sequence of the polypeptide. But under suitable conditions, we would hope that a large number of these fragments that could be generated will indeed be observed in a spectrum. And that is shown here on the right-hand side. So this is an example of a peptide from keratin. So keratin tends to be the most a very abundantly observed contamination. So when we handle any protein samples such as gel bands in the lab, it's very easy to contaminate this with our own skin cells, and they're largely made up of keratin. And what is shown here on the right-hand side is essentially where B and Y ions have been assigned to this spectrum. Now, essentially, in order to now use the spectrum to identify the original protein, it is critical to be able to match the observed fragment ions to expected fragment ions. And the way that is being performed is essentially through a bioinformatics process where we take the entire protein sequence database. For all of the proteins, let's say in the human proteome, we generate a triplet peptide. So we predict the peptides that would be generated after trypsin digestion. And for each of these triplet peptides, we now calculate the possible B and Y ions that could be generated. So we now produce lists of potential B and Y ions. And all of these essentially theoretical or predicted MS-MS spectra will now be compared and matched to experimental spectra obtained in a liquid chromatography tandem mass spectrometry experiment, where we combine MS1 and MS2 acquisition. So MS2 standing for the fragmentation or the MS-MS fragmentation of these peptides. And as you may notice, if you compare a large number of experimental spectra to a very large number of theoretically possible predicted spectra, it is critical to have a way to assess the confidence of a match. And that is being done with a score. And that score essentially quantifies the probability of that match between, being a non-random match. And that is shown over here. So this particular match here between this sequence and the experimental spectrum is on the one hand highlighted by the red numbers. So each of the red numbers in this table is a fragment ion that was actually experimentally observed in the spectrum. And what it shows to you is that actually we've seen a lot more Y ions than B ions. And that's actually quite a typical situation for triplet peptides. So trypsin is a protease that caps at the C terminus of lysine and arginine, or K and R. And these are two basic amino acids. So they tend to be very good at retaining a positive charge. And for that reason, the C terminal fragments, the Y ions, they tend to fly very well on the mass spectrometers, whereas the B ions may not ionize as well. Another reason, a potential reason, is that the B ions, as we'd seen on the previous scheme, may undergo further degradation to A ions as well. And what we can then also show in the diagram below here is that essentially the match between the theoretical or expected mass and or the mass area that can be calculated from that tends to be systematic in some kind of way. And it sometimes can depend on the mass analyzer. So the data shown here is actually from an ion trap mass analyzer, where we see that the mass accuracy is lower in the low M over Z region at low M over Z values, and tends to be better here at higher M over Z values. Okay, so just to show sort of an experimental example again, from the same study where we shown the MALDI data earlier. So again, we're interested here in the conversion of some kind of substrate polypeptide by the aspartate hydroxylase to the hydroxyaspartate or hydroxyasparagine peptide. And what we were specifically interested here was to exactly map the modification sites within the polypeptide. And what is highlighted here is essentially the B and Y ions that were absorbed in this spectrum, and in red are all of the ions which essentially help us to gain some confidence in the actual localization of the modification side. So what we expect now is that the modified aspartate residue here will have a mass addition of 16 Daltons. And those 16 Daltons have to be present in any fragment ion that contains the aspartate residue. So in all of the B ions here that contain the aspartate residue, so all of them except B2, we've seen this mass shift of plus 16 Daltons. Whereas when we look at the Y ion series, none of the Y ions except Y18, except the one that has only lost the C-terminal cysteine residue here, none of these other than the Y18 ion actually cover the region containing this aspartate on this after the conversion hydroxyaspartate residue. And of course, this can be annotated manually, as was done on this spectrum here. But generally, to go back to the previous slide, generally, this is something you would want to do computationally in the way it was described here. And one way to do this is this MASCOT search algorithm that will match the data from the protein database to the experimental spectrum. So then, yeah, coming to the end of the lecture, I just want to point you to some further reading. So here's some examples of good introductory textbooks, both on biological mass spectrometry, but also here one on proteomics. And then I would like to end by acknowledging the members of the MRC LMB Mass Spectrometry facility. So we have Farida, and here's a few of the application areas that she tends to work on. Sue, who works quite intensely on a method called isobaric labeling proteomics or TMT-based proteomics. And I will cover this in the next lecture. And then Thomas, and he's joined us more recently, working on structural proteomics, both on hydrogen-deuterium exchange mass spectrometry, as well as cross-linking mass spectrometry. And then given that I've shown data as well from my previous position, I want to acknowledge Alex Montoya, Georgia Rumiliotti, who both work on proteomics in the facility at MRC LMS in London, as well as Joao Mokoczynski. He works on metabolomics, so analysis of metabolites by mass spectrometry methods. And yeah, thank you for your attention, and I'll be happy to take any questions that you may have. Okay, I should probably point out, if you've got any questions at a later stage, please feel free to email me, and my contact details were at the introductory slides. And equally, if you watch this lecture online at a later date, yeah, feel free to contact me in case of any questions, or if you would like to discuss yeah, working with the LMS facility. Yeah, I do have a question, so I'll read this out. So the question is, how common are these deamidation reactions, and how does this affect peptides identification? So that's a very good question. So deamidation is a reaction that can occur spontaneously, essentially in the sample preparation. It is somewhat sequence dependent, actually. So you tend to observe deamidation much more frequently on asparagine residues rather than on glutamine residues. And that involves the reaction mechanism that happens. Now, it's not a huge problem, because the slide where I covered the matching of theoretical spectra and or theoretical fragment ion masses and the observed masses in the spectrum through the MASCOT algorithm, you can allow variable modification. So for any known modification, you can allow this, where you say, okay, I would like to consider for any experimental spectrum that residue X, let's say this asparagine residue, could be deamidated. And in the case of deamidation, that leads to a mass increase by 1 Dalton. And you can allow that in the search, and you will then be able to match experimental spectra that contain the deamidation to the spectra that allowed for that. It's just worthwhile keeping in mind that whenever you add these variable modifications, you increase the search space. So for every peptide now with an asparagine residue, you now need to consider that that peptide could be in the unmodified form, or the peptide could be in the deamidated version. And in that case, it means that we've increased the search space. And that will have an effect on the kind of cutoffs, so which hits still turn out to be significant or not. But the rule of thumb is sort of 2 or 3 modifications should be fine. Beyond that, it may be worthwhile considering a different type of search strategy. And again, I'd be happy to discuss these, such as open modification searches. Yeah, thank you very much. So if there's no further questions, then thanks again for your attention, and I will close this lecture. Thank you very much.